Welcome to the problem-solving set on transduction. The data below describes the number of transductants with given phenotypes, and they're asking us to determine the order of the genes. What we're going to do is look at the phenotype that has two wild-type markers, so either B plus C plus, A plus B plus, etc., and see how many transductants they have with that phenotype. So we're looking for what they're called co-transduced. Okay, so B plus C plus has 510 transduction. A plus B plus has 489. This third one, only one marker was transduced, so we're not going to worry about that for now. And then finally, A plus C plus has 6 transductants. So as you might have gleaned from the other problem-solving sets, the closer two genes are, the more likely they will be to be co-transduced. So as you can see, A plus and C plus only have 6 transductants. So we're going to assume that they are the farthest away from each other. A plus B plus has 489 and B plus C plus has 510. So they must be close together. So if A and C are far apart, that means that B must be in the middle if both A and C are close to B. Okay, so the order will either be ABC or CBA, depending on which way you're looking at. Okay, so A and C are farthest apart with only 6 transductants, and A and B are close together, and B and C are close together, so it would account for the increased number of co-transductants. So part B is gene B closer to A or C. So we know that B is in the middle, but which one is it closer to? Well, 510 and 489 are very close, but they are slightly different. So you see, again, the higher the number of co-transductants, the closer the genes are together. So 510 being greater than 489, we know that B and C are closer together than A and B are. So the answer here is that B is closer to gene C. Okay, it's very simple. Just another application of the closer two genes are together, the more likely they are to be donated to the recipient cell either through conjugation, transformation, or transduction. Problem 2. A recipient strain of E. coli with the genotype mutant for U, V, W, X, Y, and Z is transduced with a donor strain wild type for all 6 markers via a generalized transducing phage. The table below shows the results of the recipient culture being plated on media with various supplements. Note that a mutant allele for U indicates a requirement for U as a nutrient, etc. And it's asking us what is the order of the genes. So the data shows that in each experiment, 4 supplements were added to the minimum media. Now, because there are 6 markers, the key to notice is that when 4 compounds or supplements are added to the media, the other 2 are not. So first I want to go down the list and see which ones are missing from each media. Okay? So the first one, the missing supplements are X and Y. And I'll draw them just to the right. Then you're missing WY. Then W and X. V and Y, V and X, V and W, X and Z, U and Y, U and X, and finally U and W. Okay, so if you're missing these 2, I'll draw a minus so that we know we're missing these. Remember that the recipient strain is originally mutant for all 6 markers, which means auxotrophic, and therefore cannot grow unless either given the supplement of that media, such as if it's missing U, if it's U minus, then you have to give it the supplement U, or another way for it to grow is if it received that wild type U allele from the donor strain. So if you're missing 2 supplements in each of the experiments and yet you still see growth, that must mean that the alleles for both compounds must have been co-transduced, right? If they can still grow if they're missing W and Y, that must mean that they must have gotten wild type W and Y from the donor, and therefore we're going to use the theme again that if two alleles are co-transduced, that they should be relatively close together. So let's look at the ones that had growth and see which supplements were missing, and therefore which alleles they must have gotten the wild type version of. Okay, so let me draw down here. These were the co-transduced markers. So the first growth is when they're missing W and Y, so they must be wild type for now for W and Y. And therefore W and Y must be relatively close together. Then you have X and Z. Then U and Y. And then U and X. Okay, so because these markers were co-transduced, then each of the pairs must be relatively close to each other. And they're asking for the order of the genes, which is why we're looking at this. Okay, so let's draw a preliminary order of the genes. So we know that W and Y must be close together. And if you see, none of the other markers are close to W, so we can assume that W is probably towards the beginning or towards the end, whichever way you're looking at it. Okay, but then Y is also close to U, so we can put U on the other side. Then U is also close to X, so we could put X on the other side of U. And then X is also close to Z, so we could put Z on the other side. Now these are only 5 markers, so you want to also take notice that marker V is missing, and therefore it probably lies in the region of the chromosome that is at a distance to all the other markers. Okay, so really the order of the genes, we can put V at the end after Z, or we can write this order over, W, Y, U, X, Z, and put V in front of W. Okay, so this is either one is the order of their genes. We don't really know where V lies because it wasn't co-transduced with any of the other markers. Okay, so either of these, you can read them forwards or backwards, could be the order of the genes based on this data. Problem 3. Contrast generalized and specialized transduction. So generalized transduction is when a random portion of the bacterial genome is packaged into the phage and then transferred to a recipient cell. So random portion. And specialized transduction is when a prophage excises itself inaccurately from the bacterial genome, taking some of it with the bacterial genes with it. Okay, so this is when prophage excises inaccurately. And in this case, only the genes that are near the integration site of the prophage can be transferred. Okay, so bacteriophages that go through a lysogenic life cycle are the ones that can accomplish specialized transduction. So an example would be the lambda-phage. Okay, because the lysogenic life cycle is where the bacteriophage infects a bacteria, and then the viral DNA is encompassed into the bacterial genome as a prophage, and then later it excises itself to make new phage particles. Okay, so when it excises itself inaccurately and takes some of the bacteria, the DNA bacteria with it, that's when you get specialized transduction.